My lab is studying epigenetics, a field which deals with changes to the genome, that is the DNA and its organization within the cells, but not changes to the DNA sequence itself. So everything on top of the DNA epi is over. So it includes most notably chemical modifications to DNA or to the structural proteins, namely histones, that organize the DNA in living cells, as well as the composition itself of the histones, which can be replaced by different histone variants. In our ICRF project, for example, we're working on one of those histone variants, which is called H3.3. And this one was found to contain a specific mutation found in over 70% of DMG, or diffuse midline glioma cases, which is a rare and devastating brain cancer in children. So in a model system of this cancer, which we developed in my lab in embryonic stem cells, we characterize the immediate changes that occur once the mutation gets expressed inside the cells. So I think the most exciting thing is the unexpected. The unexpected is, what drives us, and you never really know what to expect, and every discovery leads to a new direction and new directions. In this project, for example, we actually generated stem cells which express the mutated form of the histone variant H3.3, as I said. So this is the mutation that is found in patients. And this mutation was known to lead to the inhibition of a specific enzyme. And it was assumed always that this is the cause of the disease. And we actually generated as control cell lines another mutation in the exact same location in the gene, in the H3.3, which does not inhibit the enzyme. So we always thought that this would just serve as a control, but when we started doing the experiments, we found actually that many of the changes that we observed in the actual mutation was also shared in the other control mutation. And this actually suggests that at least some of the cancer-related phenotypes that we are directly observing is not due to the inhibition of this enzymatic activity, but to something else. So these cancers are incurable and deadly, with a life expectancy of maybe between 1 and 1.5, maybe 2 years. And current therapeutic directions, which are all pretty novel, concentrate on the inhibition of this enzymatic activity, which I just described. But I actually hope that our study will open new directions, for example, for combined therapies, such as inhibiting this enzymatic activity together with drugs that would, for example, target stem cells, because we actually found that these mutations cause the cells to become more stem cell-like. So I think it's now really clear that not all changes can be ascribed to the inhibition of the enzymatic activity. So this is one of our hopes. So actually, ICRF funded one of our earlier projects more than, I think, 12 years ago, and actually enabled us to work on reprogramming mature cells to pluripotent stem cells, or induced pluripotent stem cells, as they are called today. So pluripotent refers to their potential to generate all cell types. And this field we're actually currently still pursuing. So it was extremely important for my lab, for my career, and the trajectory actually of my lab that we ended up pursuing. And this would not have been possible without this early funding more than 12 years ago. And actually the current project is also not the main focus of my lab. So really the ICRF grant strengthened this direction and enabled it. And it really now ensures our continued efforts on cancer epigenetics.